Hi everyone, today we will discuss about the topic electrophoresis. So basically electrophoresis is a separation technique where we separate the biomolecules based upon their charge, shape by applying the electric field. Okay, so usually this technique is used to separate the amino acid, protein, nucleic acids like DNA, RNA. So basically the biomolecules we will separate based upon the charge and the shape. Molecules should contain either positively charged or the molecule should be negatively charged. So depending upon the charge they possess, it moves towards the cathode or anode. So we have to remember that cation is positive but cathode is negative, as well as anion is negative but the anode is positive. We are going to see that cathode here, which is negatively charged, and anode, which is positively charged. Okay, so in this technique electrophoresis, consider a glass plate. Okay, consider this is a glass plate where one side is we have the cathode that is negatively charged and one side is the positively charged anode. Okay, and in one side there will be a presence of the cuvette. So this is a cuvette where the biomolecules are filled. So here and all the whole glass side consists of agarose gel. Agarose gel, that is a polyacrylate gel. So we will use the stain for the biomolecules to differentiate the color. Okay, so most commonly used stain is amido black, and we will allow the electric field, right? This is the electric field, so electric field will pass through it. So the biomolecules in the cuvette will move either towards the positive or stays in this negative also. Okay, so if the sample is positively charged, then it won't move, it stays near the cathode itself. But if the sample is negatively charged, it moves towards the opposite, that is anode. Okay, because it is attracted by the positively charged anode. So after certain time, when the biomolecules are completely separated, we will measure this, how much of the biomolecules is separated. Densitometer. Densitometer is a device from which we can estimate how much of the biomolecules have moved after once we expose it to the UV rays. Okay, that is bromide. Lithium bromide. So the color changes. So as I said, we can separate the biomolecules based upon their charge as well as shape. So till now we have discussed that about the charge. What about the shape? So what happens is, if the sample is mixed in a combination of both the large molecules as well as the small molecules, both, then if the molecule is large, it can't move, right? But even though it is anode, it can't move. So we can't separate based upon their charge, but we can separate based upon their shape in that case. As well as we know that large molecules have a less mobility compared to the small molecules. So in this way biomolecules are separated both size and based upon both size and shape of the molecules. Also one more thing we should see, that some molecule won't stay completely in the cathode. See, if this is a cathode and this is anode, that molecules may not be filled here and here itself. The some molecules will be present here also, it means in the middle. What about those molecules which stay in the middle? So it is not completely moved towards the anode, it is not completely moving towards the cathode, but it stays in the middle, means it's a neutrally charged. It is not charged, it is not charged positively, not charged negatively. So we use this separation technique for separation of the biomolecules, chemicals, and biological laboratories. So in the pharma company also we will use this process of electrophoresis. So I hope this video was helpful. If you like the video, like and share with your friends. Also subscribe. Thank you.